Good morning, everyone, and thank you for joining us for our virtual public seminar, The Power of Personalised Medicine. My name is Bronwyn Terrill, and I have the great privilege of heading up the Garvan Research Foundation, the fundraising and marketing arm of the Garvan Institute of Medical Research, and I'm delighted to be your MC this morning. I appreciate that the latest COVID-19 outbreak has caused significant disruption, particularly here in Sydney, but also in our fellow eastern states. So it's really wonderful to see so many familiar faces in the audience, as well as some newcomers. I do hope you enjoy the presentation. I've also just received notification that our chairman, Dr. John Schubert, is in the virtual audience, which I guess means we better all be on our best behaviour. Welcome, John. Lovely to have you with us. Before we kick off, please join me in paying respects to the Gadigal people of the Eora Nation, the traditional owners and custodians of the lands on which the Garvan Institute and St Vincent's Hospital are located. We recognise their continuing connection to land, waters and culture, and pay our sincere respects to elders past, present and emerging. So, today we're going to talk about personalised medicine and the future of healthcare. As you know, every individual is unique, some of us more so than others, and we differ in our susceptibility for disease and how we respond to treatments. Personalised medicine aims to tailor treatments to achieve the best outcome for individual patients or cohorts of patients, rather than treating everyone with a one-size-fits-all approach, which we now know often doesn't work. Personalised medicine is increasingly involving genomics, the study of all the information encoded in the genome. And as we're witnessing with COVID-19, and a test developed right here at the Garvan indeed, next generation sequencing is becoming faster. And as we learn more about the human genome, we are increasingly able to provide molecular diagnoses for disease and to match the right treatment to the right patient at the right time. It must be said, however, that we are still in the infancy of what will be possible for the future of personalised medicine. Here at Garvan, our fundamental goal is to understand genome biology and its impact on disease, as almost every disease has a genetic component. Our vision is to use all the information encoded in the genome to better predict, diagnose, treat, and ultimately prevent disease. In today's seminar, our genomics educator will take you through the fundamentals of personalised medicine before you hear from two of our scientists about their exciting research projects and an upcoming clinical trial. In terms of housekeeping, we have a live Q&A at the end of the presentations. So if you have any questions throughout the presentation or at the end, please do add them into the Q&A function at the bottom of your screen. We'll do our best to get to all of you. If you have specific questions that are not addressed throughout today's presentation or via the Q&A, please submit them with your name, email address and phone number in the survey at the conclusion of the presentation. We'll respond to you as soon as possible. It's now my great pleasure to introduce our first speaker, Bronwyn Terrill. Bronwyn is a science communicator, educator, researcher and writer at Garvan's Kinghorn Centre for Clinical Genomics. She is responsible for developing strategic approaches to engaging and educating the public and a professional audience in genomics, which includes personalised medicine. She is also an investigator on national and international projects focused on genomics workforce and education, teaching with molecular animations and community engagement with genomics. Bronwyn, welcome. Over to you.
Thank you, Mara Jane. What a wonderful introduction. I'm just going to share my screen so that everyone can see hopefully what I'm about to talk about. So some of you may have actually heard me talk at the last seminar about genes, and I'm really glad to be here on a very related topic of personalised medicine. This is medicine that's customised to an individual, as Mara Jane had said, and using very detailed information about them to be able to treat or focus on a particular cancer or disease. So my aim for this morning is really to talk a little bit about what personalised medicine is, the types of information that can personalise medicine, and the applications of personalised medicine. So the other thing I'll be doing is mentoring some of the work of the different centres at Garvan that are enabling doctors to practice this medicine. So what is it? Personalized medicine has got many names and definitions. You might hear it called precision medicine. You might see any of these terms: molecular screening, genomic profiling, personalized risk assessment, targeted therapy. These are all aspects of customizing and individualizing medicine according to someone's biological information or genetic information. Personalizing medicine can happen in many different ways. The first I tend to think about is really about personalized risk, which is using detailed information about a person's DNA, their lifestyle, their environment to understand the risks of developing disease or of that disease recurring. However, a huge part of the work, and what you'll mainly hear about today in personalized medicine, is around using genomic information or genetic information to get to the core of a disease and understand the pathways that could be used to treat or to interrupt a disease as it happens. What I'm going to do is use a little bit, use one of the amazing resources that's been developed at the Garvan Institute to take us onto a deep dive into the cells. I'll start the animation. This animation was created by my colleague, Kate Patterson, who is a biomedical animator at Garvan. And what she's showing you here is cancer forming when regular cells in our body start to divide out of control. Over time, the growing cancer mass destroys the nearby cells, and it also has the potential to develop the capability to move around the body. However, many of the triggers that cause a cell to become cancerous are actually found inside the cell in our DNA. Our DNA encodes some genetic information that's needed to assemble different molecules such as proteins. So it's very important information. But unfortunately, DNA is under an enormous amount of stress inside the cell. Toxins and background radiation, shown here as light particles, can attack the DNA and cause damage to it. It can cause a break in the DNA strand and making change mutations in the DNA code. And cancer arises from the accumulation of this damage in the DNA. In a healthy cell, molecules such as the one shown, which is actually called P53, what it will do is it's activated by the damage to the DNA, and then these form part of our natural defense mechanism to be able to defend against cancer. They bind to the critical parts of the DNA and help the cells, help guide the cell's response to it. But in cancer, damage accumulates in genes that are both involved in growth and also in regulating growth. So sometimes this defense mechanism can actually be damaged and it can't bind to the DNA or rally the cell's defenses. The gene that makes this particular protein is mutated in more than half of human cancers. And this type of damage is just one of thousands of molecular mistakes that can occur in cancer. But modern DNA sequencing and other new technologies can enable these molecular mistakes to be identified and detected in cancers. We know that a cancer can look the same at a cellular level, but this molecular information gives us a new lens on cancer. It also suggests that different cancers, like shown here as pancreatic cancer and cervical cancer, can actually look different under the microscope, but the molecular pathways that are damaged in that cancer can actually be the same, which means that a drug developed for one cancer could potentially be repurposed or used in another. So hopefully that gives you a brief introduction to the science behind the concept of precision medicine. And one of the main things I want to emphasize from that video is that the information in your genes underpins all your body's working, but changes in our genes also underpin many, if not all, human disease. And all the clues are in the DNA. The reason why we need to keep advancing our medical research is that different knowledge and different tools are needed to understand different diseases. So in the last seminar I did, I talked almost exclusively about the information we inherit from our parents, that's present in all our cells and spread across the DNA in packages or volumes called chromosomes. So you can see them here and from an animation that we have up on the Garvan YouTube channel. That ideally can be used to predict a person's risk of developing a disease or of that disease recurring, so that ideally something can be done to treat or even prevent it. The DNA information can also be used to predict a person's response to medication so that their doctors can avoid prescribing something that might cause them an adverse effect, a side effect, or that it might identify people who can metabolize drugs differently and give them the right dosage from the very beginning rather than going through a trial and error. What I didn't really talk about last time was another kind of information that comes from the cancer or the damaged tissue itself. We think of that as the cancer genome or the disease genome, which can actually be quite different to the DNA in the regular cells. So on the left here, we have those chromosomes, those packages of DNA all lined up in pairs, one from mum, one from dad. But in the picture on the right, you can see a cancer genome where the DNA is broken up and rearranged and fused in kind of wild ways as the cancer cell accumulates damage. So with the information directly from a cancer cell or from a piece of diseased tissue, what we can do is have a look at targeted therapies that act specifically on the cells that misbehave or grow out of control. The information can also be used to identify and repurpose different drugs. And we can also identify combinations of therapies to treat that cancer or condition. So this type of detailed information can be phenomenally powerful. The way we would hope it would work is an individual would talk to their doctor, here in our little cartoon, and together they'd work out what the test was that they would be comfortable with having. A sample of the diseased tissue would then go to the laboratory and be looked at both under the microscope, which is important, but also information from the actual cancer or diseased tissue would be extracted, being DNA or genetic material. That would then be sequenced in the various machines we have and analysed to be able to identify treatments that would be useful in this case. Those treatments would go to a team, we call it a molecular tumor board, or a multidisciplinary team, to be able to identify what report then eventually goes back to the clinician and the patient. So this is the way that we're hoping it will work. What we're finding, of course, is that personalised medicine, as it's beginning to infiltrate healthcare practice, there are challenges that go with that. There are many things that make this tricky. We know that genes are not the whole answer, so this has to be integrated into medicine as a whole. The other thing is that genes are shared with your family, but they're also unique to you. So this makes it very, very sensitive information. What we're finding, though, is that precision medicine can use parts of that genome that are either very variable and unique to you, or they are parts that are specifically focused on the diseased tissue or cancer. And that has very little impact on a family member. So what we're trying to do is figure out how to unlock that DNA to be able to use it in regular practice. You'll be able to see that people are already benefiting from these kinds of trials. Even though precision medicine is still in its infancy, people are experiencing the potential through the massive research trials that are on, including Ellie on the left from the Zero Childhood Cancer trial. And on the right, you can find out on our Disease Dilemmas website about Catherine's story, who was diagnosed with pancreatic and colon cancer, and her involvement with cancer research and the benefits that it provided her. She tells her story on there. So to finish up, I just wanted to briefly mention some of the additional work that Garvan's doing in this area. One of the things we've been working on is how to get information to researchers, to professionals, and also to families and communities. We're working on a large trial to make sure that genetic information can be present in the cancer clinic, including these polygenic risk scores, these sections of the genome that combined can provide, can cause quite a high risk for cancer. We're also working on national symposia to make sure that the information is being shared amongst researchers and looking at health professionals' perceptions of this work so that we can work out what are the key barriers and opportunities of moving this work into practice. And finally, we've also been, there's a centre, our Kinghorn Centre for Clinical Genomics has developed a test that is being transitioned to SIDPath in St Vincent's Hospital, which will be able to provide information from hundreds of genes relevant to cancer, and that'll be available in the coming months. So to finish up, I'd love to thank the people who are involved and completely central to this research, who are our participants in our studies, our clinicians and researchers, and thanks to Kate for making that amazing animation.
Wonderful. Thank you so much, Bronwyn. Fantastic presentation. It's now my great pleasure to introduce Dr. Kendelle Murphy. Kendelle is a postdoctoral researcher in the Invasion and Metastasis Laboratory here at the Garvan Institute. She is currently studying how the surrounding microenvironment of the tumour and non-tumour cells influences the invasive and metastatic capacity of pancreatic cancer. Using a precision and personalised medicine approach, Kendelle is able to identify subsets of patients likely to respond to different dual targeting treatment regimes. This is more than one drug, improving chemotherapy and patient survival. Kendelle, thank you so much. Over to you.
Just checking I'm sharing okay?
Not yet. Perhaps try again.
We love technology, don't we?
We do.
All right.
Fantastic. You're all good to go.
Thank you. So thank you for the opportunity to present some of my work that I've been doing over the past few years here at the Garvan. And I focus primarily on pancreatic cancer. So within Australia and worldwide, the survival rate, 5-year survival rate of pancreatic cancer has remained stagnant at less than 10%. And despite the best effort of researchers and also those in the clinic, this has remained pretty stagnant for the past 4 decades. Now, only 20% of those diagnosed with chemotherapy are eligible for surgery, which means that unfortunately the survival rate post-diagnosis is at about 3 to 6 months. Now, this means that worldwide, pancreatic cancer is currently the 3rd largest cause of cancer-related mortality. And whilst we've slightly adapted clinical treatment regimes, adding on a second chemotherapy, Abraxane, to traditional gemcitabine, this has only offered a modest increase in survival, adding about 6 weeks. So clearly there's a need to develop a new treatment and to try and target this really aggressive cancer. So what is pancreatic cancer? Obviously, pancreatic cancer occurs in the pancreas. And what happens as the disease quickly progresses is that we lose the normal structure of a healthy pancreas as it progresses to full-blown pancreatic cancer. Now, what's recently become apparent and is well accepted in the field is that it's not just the cancer cells that are responsible for driving this disease and also responsible for the response to pharmacological intervention, but also the surrounds, other cells that interact with the cancer, and also the architecture, so particularly the collagen and the fibrotic reaction that occurs throughout disease. Now, this drives the cancer and also influences its response to chemotherapy. So what we do know is that within a healthy pancreas, there's very low levels of collagen and fibrosis. However, as the disease appears and starts to develop, you can see here in the bright pink that there's a marked increase in this fibrotic reaction. Now, as I said, this not only acts as a barrier to treatment, but it also promotes the development and also the spread of pancreatic cancer throughout the body. Now, what's really interesting is that if we look at surgical samples of pancreatic cancer, as we would expect to see, there's a high amount of fibrosis. However, if we then take samples from patients who receive chemotherapy prior to surgery, what we see is there's a marked increase in this fibrosis. Now, what this does is this creates a loop where the marked increase in fibrosis further prevents response to any other therapeutic intervention down the line. So what we're trying to do is part of my project is try and overcome this fibrotic reaction to see if that we can then improve response to chemotherapy. So what if we can look at this interaction between the tumor cells and the surrounding architecture, and particularly the collagen? And what if we can try and inhibit this? So the protein I've been working on is called FAK. Now, we don't need to worry about the scientific intricacies in this diagram, but what I want you to know is that we can pharmacologically inhibit this using an FAK inhibitor, which acts as an antifibrotic to try and reduce the amount of fibrosis in these cancers. But the other aspect of my project is now to look into the personalization. So as within all of us, we're all unique and all different. Every tumor itself within itself is different, but every tumor between each patient is also quite different. So if we take biopsy samples from patients and look at different molecular markers in these, you can see by the graphs that there's a wide variety of levels. Some have high, some have low. So it doesn't particularly matter what these markers are, but for example, if we take one patient who has, for example, high levels of fibrosis, they might have super low levels of another molecular marker called merlin, which I'll get to later. But if we then look at another patient who has super low levels of fibrosis, they may have really high levels of merlin, which means that looking for just one particular marker as a predictive response to a drug isn't necessarily enough. So I'm going to quickly talk a little bit about trying to uncouple this and how we can use one or two companion biomarkers to try and predict for personalized response in pancreatic cancer. So when we look at samples of pancreatic cancer in those that have a quite early disease, they still have fibrosis, but this level is quite low. If we then look at our marker for our antifibrotic, FAK, we can see a wide variety of levels. We see those with high and those with low. Now, what was really interesting is that in red, you can see in the graph down here, if patients with early fibrosis have very high levels of FAK, unfortunately, it's predictive of poor survival. So what we want to try and do is try and increase the survival in these patients. Will they respond better than those with low levels to our FAK inhibitors? So to do this, we use some patient-derived xenografts where we take tumors from patients, expand them in mice, generate cell lines, and also look at biopsies. And from this, we could identify two patient samples with very different FAK signatures. So we have one here with low FAK, which will be orange for the rest of my presentation, and another with high FAK, which will remain in pink for the presentation. Now, what we also do is to try and pinpoint and make precise treatment regimes. Sometimes giving all the drugs at once isn't the best option. We need to sometimes space them apart, not only to reduce toxicity, but also to maximize their effect. So my whole project has focused on using a priming response, where first we use our antifibrotic FAK inhibitors to try and disrupt the fibrotic reaction and the amounts of collagen in the tumor, to prime it and make it vulnerable to subsequent chemotherapeutic intervention. So what I've been doing is looking at the survival in our two different patient cell lines using this priming regime. So if we take our low FAK cell line, which we predict would be quite insensitive to our combination treatment, we see that chemotherapy alone and adding our FAK inhibitor prior to this doesn't particularly increase survival, as we expected. However, if we take our cell line, which had super high levels of FAK, we see that by adding in our antifibrotic FAK inhibitor, it significantly extends survival within this patient, highlighting the difference in two different patients and their response to our treatment regimes. Now, what's quite interesting is that we look at not only survival, but also metastasis, as pancreatic cancer is highly metastatic and often spreads to the liver. So when we look in the livers of these mice, and I apologize for the gory image, but science, what we see is that whilst adding our FAK inhibitor doesn't particularly reduce the amount of metastases in our FAK insensitive patient, it does actually delay the onset of them, which means that whilst it wasn't the greatest response, there is still something happening within these tumors. Now, what's really promising is that in our FAK sensitive line, whilst chemotherapy does slightly reduce the number of metastases, what we see is that when these mice did present with them, there was a large number greater than 20. And really, really promisingly is when we used our antifibrotic inhibitor prior to chemotherapy, only half of these mice had metastases in the liver. And even more promisingly, there was only one metastatic lesion in most of these livers, which suggests that if we can use FAK as a biomarker, we can predict patients that will best respond to our drugs. Now, as I said before, sometimes using one marker isn't quite enough, and sometimes we need to look to other cancers to find answers. So we looked to mesothelioma and a second marker called merlin. Yep, it's our little magic trick. So what we did is we read and found that merlin can actually further predict for sensitivity to FAK, such as our antifibrotic drug. So when we take our two patient cell lines, the insensitive low FAK, we see that it has super high levels of merlin. However, in our patient that had high levels of FAK and was super sensitive to our antifibrotic, it has low levels of merlin. So we wanted to further test this theory and see if we could add Merlin as a companion biomarker for response to our antifibrotics. So if we take our previously insensitive cell line, can we try and make its Merlin signature look like our sensitive cell line and make it respond to our drug regimes? So we did a little bit of black magic here and we removed Merlin from this particular cell line. And what we already know is that in this particular cell line, by adding our antifibrotic FAK inhibitor, it has no effect on tumor survival. However, if we take our little black magic cell lines where we've removed Merlin, what you can see here is that all of a sudden, when we've removed Merlin, added our combination and priming treatment with the FAK antifibrotic, you can see here that this tumor is now considerably smaller than all the others. Now, I know this graph is considerably confusing, and what I want you to just focus on is this very bottom dotted green line here. So if we can try and manipulate these tumors, add in our antifibrotic in a cell line that now has less Merlin, we can actually stabilize disease. So we've taken this and taken Merlin as a companion biomarker to now further predict for sensitivity to FAK inhibitors. So if we take this approach and look at cell lines and patient samples that have high levels of FAK but super low levels of Merlin and compare these to all other tumors, we know that this predicts for poor survival. So can we then try and take our antifibrotic agents into this subset of patients and improve survival? Well, that's what we're trying to do now. And in order to do that, we use a lot of intravital microscopy, which I will let Paul explain, and also some promising clinical future developments that we are doing. So I would just like to lastly thank all our funding bodies, all our philanthropy and sponsors that have kindly helped us to do this work, and everyone else in the group and at the Garvan who supports all of us doing this research. So thank you.
Fantastic, Kendelle. Thank you so much. And I think our audience appreciates any improvements in pancreatic cancer is a huge win for the individuals affected. It really is a dreadful disease. So I'm now delighted to introduce Professor Paul Timpson. Paul is leader of Garvan's Cancer Research Theme, head of the Invasion and Metastasis Lab at Garvan, and he holds an NHMRC Senior Research Fellowship, which highly prestigious fellowship. Paul aims to understand disease in the context of the surrounding environment using cutting-edge in vivo and in vitro imaging technology. His focus is on pinpointing the molecular drivers of cancer progression and the environmental cues that cause resistance to current therapies. Paul, welcome.
Hi, can you hear me?
We can hear you beautifully, thank you.
Okay, so you may translate this for me, Mara Jean, from Scottish to English.
I can do my best.
Right. I'm not sure how I can follow those wonderful talks. Okay, can you see that? Yeah.
Perfect.
Okay. So obviously, that was a great introduction to personalized medicine. In fact, I love the introduction from Bronwyn on P53 and how mutations in that molecule can actually drive this disease. And that's exactly the model that Kendelle has actually been working on. And so it's a perfect introduction to how pancreatic cancer actually spreads throughout the body. So as Kendelle said, I'll play this movie here. So it's very easy to just simply start saying, okay, we'll do personalized medicine and we'll look at fibrosis. But the other aspect is you need to know how much of a drug to give, not just to give as much as possible, because there's lots of toxicity. So one of the things we do at the Garvan is we merge technology from personalized medicine to our advanced imaging capacity. And if you add all these technologies together, you can see what others can't. And so here you can see a movie deep within the pancreas where you can actually see blood vessels in red, collagen in purple. So that's that fibrotic dense tissue that prevents the drugs from penetrating into the tumor and getting full response. And in green, this is the pancreatic tumor cells themselves. So we actually adapt all the technology Kendelle was talking about, but we actually watch how much of a drug can we give to break down that fibrotic reaction to target that FAK as well, or FAK, as Kendelle discussed, and say, can we break that down to allow enough drug to come in, but also keep the structure of the pancreas so that the pancreas still functions. And so essentially, a second tool we have is where we can actually watch and use biosensor technology deep inside that pancreatic tumor again, and we can watch how the cancer is actually responding to the tumor. So again, this is almost like a traffic light where you can see the movie on the top. Cells without any treatment are red and green, so some cells are cycling and actually starting to divide, whereas in the bottom, when we give chemotherapy, you can see many of those cells switch to a green color and are actually arresting and they're therefore not dividing. So this is almost a surrogate marker for how drugs respond. But again, we're not doing any guesswork anymore. We're actually looking deep inside that tumor and actually watching what is happening in real time. Okay, so another biosensor that Kendall's been working on is to target that molecule FAK or FAK. And again, you don't have to pay much attention here other than when things turn from green to blue, that means that our drug is doing exactly what it says and it's switching off that molecule, which sometimes sounds obvious, but we actually have to watch how long it switches off so we actually know when to give the molecule again or that inhibitor again. Okay, so this is a picture on top that I showed earlier where you get normal vascular, normal ECM, and on the bottom we can actually disrupt the extracellular matrix and we can also disrupt the vasculature. So you can almost imagine then that the tumor doesn't have as much to feed off and those secondary sites may not be able to actually grow. So we can watch what happens in the liver as well as the pancreas. Okay, so Kendall had actually set up these experiments, so I'll click back, where you can actually prime that matrix so that it all breaks away, that surrounding dense tissue or scar tissue, and then you can actually get improved delivery of the drug. And so again, here you can see on the left-hand side, you can see that highly dense matrix. We now take it away. We don't take it completely away, we just reduce it. And now what we find is that previously when the cells are red and green, and this is like a traffic light scenario, they now switch to green. And so we can actually keep on giving this drug and eventually we start to get real responses and start to improve chemotherapy. It almost sounds counterintuitive to say, don't give someone chemotherapy, but this is a new way we're approaching this disease where we say, break down that fibrosis or scar tissue, improve the therapy response. Okay, so again, you can see if we look inside those tumors, you can see red and green cells, and again, you can see the cells switching to green, and they almost look larger because they're actually dying. So Kendall's also done this in the liver. You can see these movies. I hope these are coming across well, but again, they're a little bit gory below, but if you look at the movies above, you can see that three-dimensional movie of collagen where you've got the cancer cells green and red. And then when you actually look in the combination therapy, you've barely any tumor cells left, and they're all turned green, meaning they're really responding to that chemotherapy. Meaning the next round of treatment, we hope that they'll actually respond even better. So this is really exciting for our group, and again, for a disease that's so vicious. Okay, so when we were doing this work, a company called Amplia, which is based here in Australia, heard of our work, came and discussed with us and said, could we work in parallel, look at your data and say, can we work with you and test our drug? So they've got a drug that targets FAK or FAK. And so the drug that we were using was a proof of principle drug. And they said, what if we do some work with the Garvan in a collaborative manner to do some key experiments? This would help us take our molecule into the clinic. Now, that's a dream come true for any scientist, especially someone like Kendall, who's worked on this project during her honours, her PhD, and now part of a postdoc, which is a true devotion of 8 years. But again, 8 years from a concept to now having a company coming in and saying, we love your technology, we will use this and take it further. So Amplia worked with us. They've now completed the phase 1 trial in healthy volunteers for toxicities, and there's no toxicity issues. And now based on the data that we'll show you a little bit here, we're now moving towards next year, trying to actually put this into the clinic and actually treating pancreatic patients, which is an ultimate goal, as I say, for Garvan, for our group, and for any scientist to actually go from bench to bedside, and especially in that personalised manner. So I'll quickly go through here. We used our drug, we looked at fibrosis, and it was an antifibrotic, so that's wonderful. Again, what we thought would happen is actually happening with a clinically relevant drug. It reduces the growth in these tumours, again, these very, very vicious pancreatic tumours. And again, we can improve the response to chemotherapy. And so what we're now doing is actually using Kendall's technology where we're looking at Merlin, for example, and various other biomarkers to say which patient would respond best to their inhibitor, which molecular profile will tell us how to actually give this therapy. So it sounds very simple at this back end, but it is all that basic science beforehand that allows us to actually really educate, guide, and be part of a clinical trial, which again is that bench to bedside dream that we all have. So I'll leave it there because I think all the other talks introduced everything. And again, without the funding, without the philanthropy, without all of our wonderful funding, we can't do what sounded crazy before, and now it doesn't sound crazy. So I'd like to thank all our funding, especially our philanthropy in Suttons, Len Ainsworth, and recently ACRF, who have now funded an ACRF Insight Centre, so we can look even deeper into tumours. So thanks to everyone that believed in us, and happy to take questions.
Thank you so much, Paul. Really inspiring presentation. Paul, if I can invite you to stop sharing your screen, and we'll get Kendelle and Bronwyn to come back in to join us for the Q&A. So, special guests, I think we've got about 15 minutes allocated for questions. We do have some already coming through, which I'll prioritise. Before I do, though, I just wanted to acknowledge that I've been notified that we've got a special visitor in the Zoom room from Bangladesh, a fellow clinician who's joining in. So, you know, whilst we would far prefer to do these seminars in person, it does demonstrate that you can really, you know, provide opportunity for international collaborators and people living in other parts of the world who are interested in our work to come on. So hello over in Bangladesh. Thanks for joining us. So to the first questions, Paul, I will ask you, please, are pancreatic cancer patient samples taken while alive or only post-mortem? So we're talking presumably human, but perhaps for the mouse models as well.
Yeah. So yeah, that's a great question. So the samples are actually taken live, the majority of the samples that we have. So originally the APGI, which is the Australian Pancreatic Genome Initiative, we actually get surgical samples. So we actually take those live samples, and those samples are then shipped to the Garvan from Royal North Shore, the majority of them, and are actually grown here in the Garvan within approximately 1 to 1.5 hours from surgery. So that's quite amazing transition, in fact. And we've actually got, again, philanthropy to allow us, we actually have a WhatsApp where the surgeon will tell us on WhatsApp there is a sample, and then half of the team will actually jump into a car. We've already got a kit ready, and we're already across that bridge, ready to take those samples and take them back here. So it's a large team effort. It's not as easy as I make it out, but at the same time, having those samples, we can take a part of the sample for genomics, a part of it for growing, a part of it for experiments, and we have an entire process set up for that. There are opportunities for postmortems, and that's something that people are looking at, because in that scenario, you can actually have healthy tissue, whereas, for example, in the normal surgical setup, the majority of sample you're going to get is that tumour that they've already surgically resected. Does that make sense?
Yeah, no, it does. Thank you, Paul. And just a little plug to the Suttons. I understand you and the team use a Sutton's car to get across to get the samples, which is just wonderful.
Beforehand, we were using Uber, and so I didn't think it was appropriate to use Uber to do such a thing.
Transporting human samples, and that call can come at any time, day or night?
Yeah, so the guys, as you can imagine, most of the people are Kendall's age, so they have a social life, and they're not very happy to go over on a Friday night. A lot of the samples were taken on Fridays. Wednesdays from the public, from the private, typically happen on Wednesday also, but quite often there's lots of surgical samples even on weekends, and you cannot predict that. But that's the devotion from this team, and that's why we have a rotation, so that someone does do it once or twice, and then it's someone else's turn, because again, we can't predict it.
Thanks so much, Paul. Kendelle, I'm going to ask you the next question. What causes or is the process that creates or promotes the fibrosis that you're talking about in the microenvironment? And is it unique to pancreatic cancer?
So I'll answer your second question first. So no, it's not unique to pancreatic cancer. It does occur in other cancers, breast cancer, for example. It does have not necessarily more, but it has an adaptation to this matrix, which is in a lot of other cancers. And what causes it? Well, that's a huge debate in research. So there's many drivers, not only from just the cancer cells, but also some in the surroundings. So one of the major cell types that's in the surrounding is called fibroblasts. And even within this particular cell type, there's all different subtypes of these cells. And there's one subtype in particular that's known to particularly drive it, to produce it. And then, of course, you also get more feedback from the tumor, and they just keep interacting with each other and driving and driving. There is some debate if it comes directly from the cancer cells, but most of it is from these fibroblast cells. And yeah, it just keeps driving and driving until there's so much that it becomes a literal barrier.
Fantastic. And in Paul's talk, he said that you're using these treatments to dissolve a lot of the fibroblasts, but you don't want to take it all away.
Yes.
Can you explain why?
So there was a lot of research actually where we're just going, let's get rid of everything because we know it's bad, so let's take it all away. And instead of giving the results that everyone thought it would, in that, okay, now we've fixed all the problems, it didn't. It went the other way, and the tumors actually grew and spread and developed even quicker than if you didn't touch it. So it's not necessarily a good thing to completely eradicate it. We need to manipulate it in our own way to modulate it. And so that's why with mine, we didn't completely remove the collagen and the stroma, the fibrotic reaction. We just sort of lessened it quite a lot. And that means that the tumor is actually stalled. It's not free to escape. It's still stuck there and rendered vulnerable to chemotherapy.
Fantastic. Thanks for that. Before I go to our next question, Bronwyn, can you tell us a little bit about how genomics has changed during your time working in this space?
I'm about to give myself away that I've been working in this space for some time. So back in the 90s, we didn't even have a map. I've been talking about genetics and genomics for more than 25 years, and it has changed phenomenally. It's become digital. More than one sample. It took us a long time to get the first genome and the first set of information, the first map, and it's taken us really up until now, and will continue to be able to document the variation that's going to make genomic information valuable to individuals. So the infrastructure has changed, but what it's meant is that we can ask better questions.
Fantastic. Thanks, Bronwyn. I'll go to the next question. So how to compare between chemotherapy and sensor therapy for recovery from pancreatic cancer? Which one is suitable to recover pancreatic cancer? Paul, is that one something you'd like to endeavour to answer?
Yeah, I'm not quite sure what that question is, but I'll attempt to answer that question. I think what we're getting here is one biosensor reads out chemotherapy and the other biosensor reads out that stromal response. And so it is a good question, such that when we start seeing that transition, when cells become green and the cell cycle stops and the cells don't move, we actually have to correlate how much is enough to break down that barrier, just as you were saying before, and actually allow enough drug in where it's not too much. Because otherwise, one thing we didn't mention is that if you break down that matrix far too much, you can actually get almost an opposite reaction where the cells can now start using it as almost a super highway when they can start to spread. So for many years, again, I think I mentioned this, I was even laughed at with our colleagues saying that's not going to work. And again, that's what's wonderful about science. You have to actually go in and say, it doesn't work if you smash it, but it does work if you just manipulate it. And again, that's where we believe that imaging can tell us those subtle changes or those nuances that actually respond better. So does that make sense?
It does. Thanks for that, Paul. Kendelle, what inspired you to come to Garvan to do your studies here and to stay on
So I actually did my undergraduate in Western Australia, and I came to the end of that and I thought, okay, where to now? And I was always fascinated, particularly not in actually the stroma, but more metastasis and trying to stop cancer spreading. And I just sort of started looking into different labs and stumbled across Paul and found his papers and what he did in the lab. And the microscopy was just absolutely amazing. And I didn't believe or know anything was possible before that. So I came as a little naive honors student and still here. But I could see where the project was going and the potential for it. And now with the collaboration with Amplia, it's just every sort of day of hard work or every weekend that we do, the fact that we can now actually go into the clinic and have a real impact and potentially improve survival of people in this disease. I never thought I would see something like it in my whole career, and to see it not long out of a PhD is just, I still sometimes pinch myself. And yeah, it's just to be able to have that type of impact on someone's life, and the collaborations here is just amazing. So that's why I'm still here and will be for a while.
Congratulations, really, it's quite phenomenal. Bronwyn, one for you, please. How does the world coordinate genomic studies so that, one, information is shared, two, not all are reinventing the wheel, or is everyone doing similar work actually more ideal?
Ooh, I think Paul could probably comment better on the people doing similar work actually more ideal. But I guess one thing to say is that science is enormously collaborative, and particularly in genomics, because once there was an opportunity to be able to share information that didn't necessarily involve sending tubes around the world or sending hard drives around the world, it meant that you can have maps that are shared and mirrored at multiple sites all over the world. And I think there are now huge collaboratives of working on how to make the most of the genetic information, because people are precious and samples are precious and information is precious. So the collaboration that's going on in genomics at the moment, particularly, is unprecedented, and it is really important. But I'll leave you, Paul, you might want to comment about, is it useful to have people doing the same thing in different places?
Yeah, thanks for those questions, Rose. I think Rose should probably join our group. She's got more ideas than I've got. Rose, you're absolutely right. So Kendall's work took 8 years, 60 people on the paper. The APGI is on there, a consortium of 120 people. And we've got the ATMA, which is the matrix equivalent of the APGI, which is about 60 people or so. So ultimately, we have people from practically 15 to 16 countries, I think, on there. And so we all have to work together. The APGI actually gives out, and it's based here, but it's a national infrastructure of biobanking, live biobanking of these samples. Because when you have an idea in a mouse, you then actually take the patient and you can say, how does this work? And those samples, we have emails and requisitions to actually take those samples every week. And I'm part of that executive, and we approve either a tiny slice for people to test their idea, and then we actually send the cells that Marina Pajic and our group have just started making new fibrotic versions of what Marina already has. These get spread to the rest of the world. If we work as a unit, we actually get this disease, which, to be quite honest, is quite embarrassing to say that it hasn't changed for 4 decades, and that needs to change in my career. I don't have as long a career as Kendall, but I still have 20 years to give it a go.
Thanks, Paul. Tell me, is it true that the APGI consortia was the first time that we understood the genetic heterogeneity of pancreatic cancer? Perhaps you can talk a little bit about that, because if I understand correctly, that's one of the reasons why pancreatic cancer is so particularly hard to treat.
Yeah, I worked here before that actually happened, but my plan was to come back here. And again, it was, you talked to Kendall, why did you come here? That was one of the reasons why I came here, because we could actually for the first time see the genetic changes. I think Kendall talked about it in Bronwyn as well. The one drug fits all. It's just not going to work in this disease. And there's almost, currently we'll say there's 10 versions. There probably is 12, 14, 16 as we change. But if we think for the last 30 years, we were at 5%, and we can now say, right, there's 5% that have actually got for example, BRCA mutations, and they can't repair their DNA, just as Bronwyn beautifully showed. If we attack that little cohort, we've changed that 5 to 10, and then there's another 5, and then we make it 15. So you can see what I imagine I want to do in the next 10 to 15 years, which is I will take each of those hits, and eventually we won't be at 10%. We'll be at 20, we'll be at 30, we'll be at 35. So that's the beauty of knowing that information. And now what we need to do is really go after that personalised approach and say, we need to stop doing that. And again, I think Marina talked about it before. We've got links with Nectar, we've got links with the most for 14 different sites. And that also matches those small subsets and those small genomic changes to drugs. And again, the work that we're doing with various other companies as well does that. And that's why I don't care which company or who I work with. I just look at it individually as little Lego pieces. And overall, this disease needs to change.
Thanks for that, Paul. I think we've only got time for one more question. You've touched on the importance of the pharma relationship for this particular work with Amplia. I think sometimes the community thinks that pharma are, not research friendly and not prepared to get involved with research. My understanding is that the landscape is changing dramatically because of genomics. Can you talk a little bit about what the relationship is like and how critical it is to actually build those commercial partnerships in order to do the clinical translation into patients?
Yeah, that's a great question. So, big bad pharma, right? It is the opposite now, actually. So I believe that previously experiments were done and it was like, we're working a little bit with this company. Now you work as a collaborative. They do not have the technology that we have. They can't afford to do those details. And even when they come in and saw, for example, Kendall's work, they were like, we cannot afford to do that again. What are the key experiments that are going to make the difference for this drug? And so we work as a collaborative and as a unit. And again, there's other companies out there that are trying to target FAC, but it's not until they can see how we've approached this as a priming mechanism that you can minimize the toxicity, get the exact same response, if not better. And Kendall has shown that less drug is better, which is amazing. So we're working with various companies and they really do come on board. So other companies that we've worked on, we will get a grant, they will give us the drug, but then we say, we would love to give this with immunotherapy, we'd love to give it with this, but we don't have the money. So then later can say, well, this is a win-win situation. If we're going to open up to Australia and we're part of global networks for different trials, they will open up this country and they will say, we will test a combination therapy and we will pay as much money as you pay, we will pay this. And so it is becoming a partnership and I'm enjoying it because I'm not going to spend the rest of my career just publishing papers because everyone thinks academics just publish papers and everyone thinks pharma just want money. It's actually not the case. We have to work together.
Fantastic. Thank you so much, Paul. I'm going to get in trouble, but I'm going to ask one more question of Bronwyn, if I may, because Paul's touched on the importance of the pharma relationship. We've got a number of incredible donors and advocates in the audience. Can you touch perhaps on how important philanthropy has been here at Garvan for our genomics capabilities? No need to go into specifics, but what have we been able to do that we couldn't have done without philanthropy and without the Garvan family?
Oh, everything. In reality, genomics is expensive and it has been expensive. Any emerging technology is expensive. And I think the great thing about that Garvan has been on the front foot since we've been able to really do personal genomes, individual genomes, and philanthropy allowed that movement from what was effectively a small boutique, this is what we could do, small research, to industrial scale. And that's what has enabled a huge amount of the genomics movement in Australia. So philanthropy has been absolutely vital to that. Fantastic.
Well, Bronwyn, Kendelle, Paul, thank you so much for your incredible presentations. It's really a thrill to have been able to hear from you today. Everyone in the Zoom room, thank you so much for joining us. We hope you've enjoyed the presentation. As always, we would value your full and frank feedback whether it be glowing or otherwise. Anything that we can do to improve on our presentations, on our content, on the way that we speak to our Garvan family is gratefully received. So with that in mind, there will be a survey popping up on your screen shortly. So please do take the time to give us that feedback and rate your experience for today. I'm conscious that so many of you are already donating significantly to the Garvan Institute, or your partners for the future, your community fundraisers, your advocates out in the community for our work. If you haven't already done so and you would like to make a donation, we would be humbled by your support. If you'd like to be an advocate for Garvan, please do that, and you will receive a recording of today's seminar. If you enjoyed it, please do share it with your friends and family and point them to the Garvan website, where there are a range of resources and videos where you can hear about some of our incredible patient advocates, such as our disease dilemmas heroes and many, many others. The next seminar will be scheduled soon, so please do keep an eye on our website for notification of those details, or join up to the Garvan e-news to receive notification as soon as that is available for booking. We look forward to seeing you at the next virtual public seminar. And in the meantime, we wish you and your family only the best. Do stay safe, stay well, mask up, take care. Thanks so much.